Welcome to Sungrown Stories. I'm so excited to have you here, Dr. Gary Yates. Welcome to the show. Can you just give a little bit of a brief introduction of who you are and what you've got going on in the cannabis space?
Sure. It's a pleasure to be here, Alex. Thanks for the invite. I'm sure we've got some interesting stuff to discuss. But yeah, so my background, given the doctor title, is an academic one in many respects. But my relationship with plants goes back way beyond that. I grew up in a household with a very avid gardener, and that basically instilled the principles that have guided me all the way through to here. And actually, some of those core principles are still as true today as they were when I learned them at ages, 7 through my teens. But yeah, so let's go a little bit. That's kind of starting out in reverse order. I am doing a bunch of projects just now, which maybe we'll go into in more detail shortly. But I recently just finished up my employment with Seedsmen / Pharmaseeds, which was a kind of 3 and a bit year project, I guess, or job where we were improving the genetics, improving the quality control, trying to bring a bit of science to the approach of all aspects of the genetic supply. Meanwhile, we were trying to serve the trying to give genetics into the legal industry in Europe, the medical industry in Europe. So that was a bit of a hit and miss in terms of the industry wasn't really ready for that level of specialist, I guess. Here, whilst the stuff we were doing in Seedsman was actually much more interesting and much more liberating, because we got to the biggest customer bases in North America, where many laws are less strict than they are in the UK right now. So that was from before that I was studying an algae at York University. We were looking to basically reverse engineer this algae's ability to concentrate carbon around its sites of photosynthesis, which basically made it supercharge photosynthesis rates. But really what it was a mechanism to deal with a low carbon environment, which happens sometimes in water cycles where these things live. That was really fascinating. Learned a lot about photosynthesis, light capture, light receptors, things like that. Prior to that, I was basically doing my PhD, where my thesis was based on plant pathogen interactions. So we had discovered on the bacterial receptor FLS2, we discovered a little signaling molecule called SUMO, which was activated really early on and after detection of bacteria. We discovered a class of enzymes in Arabidopsis, and during that time I worked tissue culture on rice. I did various crop trials on wheat, and worked on big crops like that. And then prior to that, I did just a straight bachelor's degree in genetics, general genetics, not plant specific. And then I had a bunch of weird things I did before that. I was a musician for 3 years and full time, and then I was what else did I do? I was a salesperson for 4 years. But yeah, that was me basically just finding my feet in life before I fell in love with science, really, and that's what really brought me here, where I am today, I guess.
Amazing. So, how did that connection though with cannabis? Was that just like natural? Have you always smoked and enjoyed cannabis, or how did you make that transition from academia and conventional agriculture to cannabis industry?
Yeah, it's a bit of a strange one. It was sometimes it sounds like a really clear path connected all one after the other, but it was much more, I guess, random than that. But growing up where I grew up in West Central Scotland, it was hard to avoid cannabis. It was pretty much everywhere. It was widely used, but in a much more abusive way than maybe we would describe now, because people were using it to get out their face, in terms of they would drink alcohol, they would take pills, and they would smoke weed all at once. And it was very much a kind of toxic cycle. They would also mix it up. It was always hash and tobacco. That's what we're known for in the UK a little bit, tobacco joints. But so that was my my first experience of it. But it was interesting because even in those early, not that I was taking all those other things, but just just the cannabis itself was something that helped me focus my mind. I found it, I had lots of ideas all the time, and it helped me really just pick one and get to the end of that idea before I would go and explore another. At the same time, I was learning how to play guitar, so those two things went together really well. And again, not having all this background noise in your mind whilst you're trying to discipline yourself to really just sit and practice scales to a metronome, you know. So for me, it was a bit of a lock and key situation. Cannabis, just perfectly fit into my life to help me get further ahead in the things that I wanted to pursue, instead of just like looking at all the noise and just it's like, not that I want to criticize anyone, but there's all this talk these days about ADHD and things like that, where people lack that focus, and I think that if I was in that category, then cannabis was the thing that really got me out of that, and really helped me decide, pick a horse and ride it to the end, in that sense. But yeah, so that was my initial dealings with cannabis, was like kind of street cannabis, recreational, young, silly, that thing. And then later I'd spent some time in Amsterdam and got a slightly different relationship with now cannabis proper, weed, buds, flower, instead of tobacco and hash. And that was again a completely different. Whoa, is this, this is completely different. This is even better. This is all the focus and all the fun without this sitting like, whoa. A lot of that actually came from the tobacco, I think, on retrospect. But so that was again another eye opener, and it was in Amsterdam, after consuming some truffles there. Within a year of doing that, maybe aged 23 and a half, 24, something like that. Within a year of having that experience, I was studying genetics at Glasgow University, having had no right to be studying anything, because I found music late on in high school and I kind of just didn't go to exams and stuff, and I was going to be a rock star and that was it, I was already written in the stars. So I kind of abandoned all things scholar at that point. And I also never learned to study in high school. It wasn't that obvious to me that you had to put work in order to pass exams, because at primary school you were just clever, or you were in that group that needed extra help, basically, and it was funny to me. I just missed the transition between putting in the work and passing exams to just knowing what you should know. So it was weird. It was a wee bit maybe naive in that sense. But yeah, it was after that trip, I had this burning question: why does cannabis do what it does? And again, prior to studying, my knowledge was, and no other plant does that, and that must be something at the genetic level that I can go and investigate. And it was basically that single silly little throwaway thought that led me to go study genetics. And then so it's kind of like it was cannabis, it was psilocybin, and a silly idea born from both of them. And before I knew it, I was balls deep in Mendelian genetics and Punnett squares, and I loved it, absolutely loved it. Where did you lose me?
Balls deep.
Balls deep. Oh my goodness. Yeah. So yeah, balls deep in Punnett squares and Mendelian genetics was and I loved it. But the thing that really stuck out was, with all the stuff I was studying, and I did extra credit stuff, like I wanted to do ecology and zoology, and I wanted to do molecular biology, and I did it all. So I did it all. There was one point where you had to pick half your subjects, and I did over 2 years all the subjects in biology essentially, which gave me, I didn't realise at the time, but it gave me a huge advantage later, because it kept my scope of view really wide, and I didn't get caught up in the detail of any one thing. But I really fell in love with molecular biology the first time I realised that there was a mechanism that sits between I don't know if the viewers are familiar with microbiology, but some bacteria will have basically an oblong body and a little tail thing that kind of flicks to help them move along, called a flagella. And when I realised that mechanism at the molecular level was basically a rotor. Ions pass through the membrane and it turned this protein, and that caused that thing to whip. As soon as I seen a video of that, I completely fell in love with molecular biology, because all of a sudden, another kind of weird passion of mine is engineering to a certain extent. So when I seen this at the microscopic and natural level, it just, my mind literally blew, and I was all in. That was it. I was going down that route no matter what. my mind was just blown with with the engineering at a molecular level by Mother Nature. That was crazy to me, and I had no idea that some of these more sophisticated engineering feats that humans have achieved were actually already embedded in the biology around us. Like, that literally blew my mind. And even yeah, even like the kind of bring fast forward to now, the cannabinoid and terpenoid synthesis pathways and how those work, and how you have all these subunits of different things that come together, and and even just how do they come together, and it's just they just do. It's that these kind of things just constantly just kept blowing my mind, and that's when I realized that you could study plants in a much more ethical and carefree way than animal biology. That's when I really started to think, right, plants are the thing for me. And again, because of my background with my grandfather teaching me gardening and basic home growing techniques of everything from tomatoes and leafy greens to roots and all sorts of in between. So yeah, it was there's these moments of, I guess, just clarity dotted through this journey where everything came together at a precise moment, and I just knew I was on the right path. I mean, everybody's on the right path, in a way, but everything just felt like, it felt like big nexus points where all these random skills came together in one place all of a sudden. And that happened, like I said, 2 or 3 times, which is why I ended up getting into cannabis from academia, because at the time it was during the lockdown and the COVID chaos, and my position in academia, I'd been a key worker, so I was still going into the labs, and then that then, because basically had an effect on the length of my contract, which was the length of my time to finish the project. So I now had to do basically 2 years worth of work in 9 months, and that didn't really make any sense. So basically, yeah, I ended up getting in touch with Pharmaseed, who I didn't know was Seedsman at the time. They were basically a startup company in the legal cannabis space, and I had a very honest interview, and that's what basically got me in, because by that point I was, I think, 16 years experienced with the plant. So, that was something that wasn't, first of all, easy to say in an interview situation, but once it was out there, it was kind of hard to look past me in that sense, because there wasn't so many people with that level of academic achievement who also had that level of experience growing, so that was that. I think that was the thing that, again, it felt like another nexus of all these different points in my life coming together in one. And I even told the story of what happened in Amsterdam to get me to study genetics. I told that story in my interview, because why would I not? Because it
Yeah.
came from the right place. So but like I say, it's weird here because we have got so much stigma still attached to these what I call natural practices, which have been done for millennia in various cultures. And even we have a local psychedelic mushroom called Liberty Caps, and they just grow everywhere, and I find this like really weird, because if it's there, if you walk past it, it's fine, and if you pick it up, you're now charged with a Class A offence, which is just mind blowing to me. But anyway, that's the cards were dealt. So yeah, that's a bit about how I got into it, and since then I've kind of just felt welcome in the community, and I've never felt that in a career zone before, even musicianship and stuff like that. It was always, I was far too egotistical and competitive, for example. Whereas actually, if you can get past the ego growers in cannabis, there's actually the majority of people out there are actually very open to new ideas and how do you do it compared to me, because I want to learn. And that was like yourself and like the guys at Humboldt and even Jorge, who's 70 years old. I want to learn, they tell me more. And because we've been in the dark for so long, and now we have all this research coming out, and it's getting, again, more exciting. And that's what's happened, I think, over my entire academic career. But since I've become to cannabis, it's just, I remember doing a survey. Normally, I've changed subjects in academia like 3 times, and it takes you months, like 9 months of reading literature to really get to the edge of the research, right? In some of these fields that are, centuries old. But when you come to cannabis plant science, I'd consumed it all in like 90 days, You know, it was like there wasn't a whole lot out there, and a lot of what was out there was really bad in terms of how they actually approached the growing. They were trying to do experiments on the plant, and you look at the growing style and what the parameters, and you're like, that's crazy. You know, that's not how you grow cannabis. And so anyway, fast forward to now, I feel like it's just you can't keep up with it anymore. You know, it's just coming out so fast, and I absolutely love it. It's really inspiring me to, get deep on that side of things again, because now we can actually make all the connections and draw broad conclusions.
Let's take a quick break for a word from our sponsors.
Welcome to the most affordable path to organic gardening excellence, brought to you by Queen of the Sungrown. Today, we spotlight an age-old secret embraced by gardeners worldwide, now made more accessible than ever. Yucca root extract powder. Envision your garden not just surviving, but flourishing, even under the harshest sun. Thanks to yucca root extract, this dream is within reach. Derived from the resilient yucca plant native to the arid regions of southwestern North America, our yucca root extract powder becomes an indispensable ally for your garden. Why yucca? Its myriad of benefits speaks volumes. It's your garden's shield against heat stress, ensuring plants remain vibrant and productive. It's a natural enhancer for nutrient uptake, making every drop of water and nutrient count. It transforms your soil, encouraging healthy growth and aeration. And it acts as a gentle yet effective pest deterrent, safeguarding your garden naturally. The secret isn't just in the plant, but in making it available to all. The saponins in yucca act as natural surfactants, facilitating critical water and nutrient absorption, while its biopesticidal properties offer a sustainable solution to keeping pests at bay. With Queen of the Sungrown, yucca's full potential is unlocked, bringing unparalleled resilience and vigor to your garden. Transform your gardening practice with the most affordable yucca root extract powder on the market. Exclusively for our listeners, use code SUNGROWN for 20% off your first order. Experience the transformative power of yucca today. Your garden will show its gratitude. Visit us at queenofthesungrown.com to discover more and claim your special offer. Queen of the Sungrown. Harness the affordable power of yucca root and watch your garden thrive. Well, Cat Lady has a question actually for you. This is great. She wants to know more about how you can evaluate a cannabis study.
Oh, wow. So yeah, let's start with where I started then. How to evaluate a cannabis study? Look at how they grew the plants. Look under the conditions, the environment. Sometimes that detail is very vague in the scientific literature. In my experience in growing plants in academia, tells you that when you come to write up that paper, right, and you say, oh, this was the conditions over this amount of weeks, and this is what we did, right? What you don't write in the paper is we had a power cut halfway through for 2 days. We had a powdery mildew outbreak in week 6, and and all of that stuff is actually quite common, unfortunately. And sometimes it does void the experiment, but sometimes it doesn't, because maybe the output wasn't dependent on those factors. So I would look at the growing conditions for a start. I would also look at what question they're trying to answer, because sometimes this is not obvious by the title and even the abstract of the study. Like for example, I think what's largely been under-addressed in cannabis is the phenotypic plasticity within a plant. Like that micro-environment differences that cause one bud to be slightly more this than that bud, or that's not been addressed well enough. It's starting to catch up a little bit now, but that's something that I felt for the start was not addressed well enough. And in order to do a study like that, you have to grow the plants under very, very tight control. To try and replicate as a consistent environment as you can, you really have to do that. So I would look at that. The second thing that I think is a big problem is what's the N number? So what the N number is in science is how many plants were involved in the study, which you should have repeated at least twice, but preferably 3 times, right? So if the N number's 4 and they grew from seed, the natural variation in the seed is probably enough to account for any downstream effects that you found different between the individuals. Now, if your N number is 1,000, then the power of the statistics helps to normalise that background variation, and then you'll get much truer results from your study. So that's probably the 2 main things that I would look at: is growing conditions, how many plants were used in the study, also look at whether or not they had fair controls. For every aspect of something that you're looking at that makes a difference, you have to, in my mind, have 3 controls, so that if any one of the controls is a factor, you're not also accumulating that kind of error in the data. So yeah, that's just been really rigorous. When I worked in the plant pathology world, it was so you had to, every dot of every I and every stroke of every T had to be done, or you would just get rejected. You wouldn't get published. And I got quite a lot of publications at quite an early point in my career. So I got used to that rigorous methodology where you couldn't give them a reason to say your study was invalid, So and again, some I don't know, some of the journals that these studies are published in as well, they don't have a high reputation. Now, that's not to say that you don't get absolute quality research in there. You do, 100%. Where it goes in terms of publication isn't actually as big a deal as it once was. But you also get a lot less vetted stuff going through that way, And one of the things you can't find out very easily is who did the review of the paper. So if it's a cannabis study, but they've got a bunch of guys who grow corn to review it, they're going to miss some of the nuance that cannabis holds, And so those kind of things, and pretty quickly you get a picture of how valid the study is. We've seen better ones now, but largely a lot of them up to date, when I joined the industry like 4 years ago, they were largely quite poor, Yeah.
What kind of advice do you have for us? So I talked to you about the Citizen Science Project, a year ago. I approached Seedsman with this idea of collecting the data from people all over every zone. And with your scientific rigorous backgrounds, publications, studies that you've participated in, I would love for you to give me feedback on what we can do to just make it better. I mean, I reviewed all of the questions with you pretty much. That was a year ago though, but so we're going to be going over when you germinate your seeds, what you're planting in, the dates, the hardiness zone, like your zip code, I guess. So it'll give you latitude as well. It'll be like a text survey that goes out once a week, asking all of these questions. When's the first time you see like pre-flowering, noticing growth rates? Are you feeding? How often? Just a lot. It's going to be a wide range of data collection, because this is the, kind of beta test to see what's actually important or significant, and if we can see any kind of correlation with the data that's collected. I would love your feedback and opinion on what we can do and what you think that we can pull from this data.
Yeah, amazing. Absolutely. I'm all in. Like, as we spoke about a year ago, we have similar minds, because I had this idea that especially through the Seedsman network, I was thinking, well, you have the power of numbers. You have that N is huge potentially within the community, and that's what we can depend on. That will help us make the data robust in itself, because if we get lots of participants, then we have lots of N's, which means the data becomes more robust. But you've also got things like, so for example, if we could validate the variety, so if we had a way of going, okay, some of these genetic tests are getting really cheap now, so if the person who bought the seeds, okay, is this, I don't know why I always say OG Kush, but that's always the one thrown under the bus in this example, but is this the real OG Kush, or is this just some genetic seed that someone's wrote OG Kush on the bag? Because if we got the data in and then we could start to categorize into varieties, we could very quickly actually see maybe tendencies within the varieties that explain, that pre-flowering started earlier there, but only in that one case, and then we could really scrutinize the data to find out other particulars, and then, you know, you've got the climate zone, and then we say, but within the cannabis gene pool, you've also got this level of variation within that zone. You see what I mean? So we can really screw into the nuance of the detail. And then the other thing would be, again, just making sure that, again, it's a harder one, but make sure the reporting is as consistent as can be when you have so many participants, you know. But yeah, I think as the data comes in and we start to discuss it, we'll be better placed to turn the screw and really make sure it's robust. As the data comes in, basically, we'll be able to scrutinise it. It will help us decide which way we want to manipulate it to, you know, help people understand it and take it in better. And by manipulate, I don't mean make it up. I just mean how we want to sort it and, you know, shift it around so it makes a visible, you know, representation of the facts. So yeah, I'm really excited to see how it comes in, and it might take some time to refine the process as we go, you know, but that's all fair game, because I think one of the things that we spoke about last time as well was what you're doing there. You know, hopefully we can roll it out to Europe, and, you know, I can certainly help that aspect of it, and then we start to get a bigger and bigger picture. And then why not, if people in Thailand, for example, can be, you know, I've got some friends there, so that's going to be then turn into some kind of monster that, you know, I don't know how you control it at that point. But, you know, again, bringing all the data in, if we've got a process for analysing that data, and it's fairly quite a straightforward pipeline, you know, giving people access to that, or at least, you know, allowing it to help you give people advice would be huge, because that's, again, one of the other things that's missing is like that collective mindset.
So I have a meeting with Tom next week I haven't even told him, like, I told him a little bit, like, through Instagram messaging, maybe email, like, hey, I'm going forward with the citizen science project. I want to interview you. I want to talk to you. So my thought is, like, so this year, you know, it's funny you said OG Kush, because that's what everyone's growing this year.
Right.
That's what the Humble Seed Company has donated as OG Kush.
Well,
So I think we're going to have a very small pool this first time, just so that we can control and make sure that the data is useful, that people are participating. So I told him between 20 and 100 participants, and they've already sold out of the triploids, so he set aside some, but he hasn't told me how many, so I'm not sure what our pool is going to be for participants. But the hope is then I'm going to talk to Tom and say, you know, this year we're refining it. We're figuring out the data collection, the software we're using, and just get all the bullshit details out of the way. And then it would be great to leverage seedsmen with every seed purchased, like, here's a link if you want to start tracking data on this strain as you're growing it outdoors. It's only for outdoors though. So, and then you have the opportunity, and we can just start collecting this database of information on every strain that's grown and how it's doing outside in different environments. I mean, that's going to be so much data, but, like, I feel like with AI, we can, you know, at least try to get through it in some cohesive way and organize it pretty quickly we'll see, with your help as well.
yeah. I became like, before AI, of course, like, I became really good on Excel, and, you know, like, in really big data sets, which, you know, constantly made my computer crash. But you know, sometimes you've got to do what you've got to do, and, you know, I've also had to learn to code a couple of pieces of script. Just not I didn't learn how to code in general. I just learned how to code this specific thing, to help with the data sorting and handling as well. So it's something that people don't really appreciate in biological and molecular biology, is that, you know, still 50% of everything is analysing the data and making sense of it. So statistical analysis and all that kind of fun nonsense is really essential to it. But I actually don't mind maths. I enjoy it. You know, like, I find it quite satisfying to get, you know, to resolve, I guess, the point of what you were trying to say with the data. You know, if your hypothesis holds up, for example, and then you do all this calculation and it shows you, you know, p-value is over, you know, 0.5, you're like, yes. You know, it's that feeling of relief, like, yes, it's not just random noise, it's a real thing. You know, and sometimes you have to spend 3 days doing that calculation before you get that answer. But yeah, that's I think the power of that is just enormous. And I'm so glad you've taken that mantle up, Alex, and actually pushed that forward, because that's the hard bit, is actually getting it off the ground, you know. It's okay having the idea, but actually making it work is, you know, so hats off to you for that. That's an amazing and massive undertaking. And yeah, you've got my full support wherever I can to make it work. Because again, even if your first one is that, you know, less than 100 people, it's still a good end number, and it certainly gives us a way to refine the process and streamline it for hopefully more masses. I don't want to speak for Tom, but I think he would be interested in that, and that, you know, thing for his customers as well, as a way to kind of feed back into the knowledge centre. And again, he'll probably want some data points from it for himself, which is fair enough. Not for himself, but for the, you know, to show next to the seeds, for example. So that's yeah, I think that's a good idea. And like I say, quite happy to be involved in this, help in any way. So, you know, you can always throw that in the mix as well, which I'm sure. I mean, so Tom and I are friends, so I'm pretty sure that'll be good for the conversation as well. Yeah.
Amazing. Thank you. Because right now it's me and Cat Lady. So, with your background, I want to talk about germinating a seed, and I want you to share your opinion. You know, you hear all these crazy things, so people are going to be starting the seeds, and I want to give throughout the podcast like tips and tricks that are scientifically valid and not just the bro science from the entire life cycle of the plant. So this podcast will follow the Citizen Science Project, but also educate and, you know, inform. So I hear people soaking their seeds in hydrogen peroxide.
Right.
And my thoughts are, what does this do to the microbiome that is present on the seed that is passed down from the mother? Because like it's a, you know, hydrogen peroxide is going to kill a lot of bacteria, correct? It's going to be, I feel like it would be detrimental, but what are your opinions on that? How do you think the best way to start a seed is? And just elaborate a little bit on that seed microbiome if you can.
Yeah, absolutely. I'm glad, that's a good question to ask. I think like, so, okay, so my opinion's a little bit split on this, because if I'm working for a commercial licensed company to do medical cannabis growing in Europe, then we're entirely using an inert growing system, essentially, right? So everything's like rock wool and salty water. Nothing is allowed to be done in soil, for example, right? So it goes against all my natural tendencies, but I've had to learn how to do that. In that situation where timing is on the timing of a commercial grow is obviously got bigger ramifications if you mess anything up, right? So getting everything synchronized, soaking seeds is a good way to help synchronize germination. Using hydrogen peroxide there is a way to prevent any potential early infections that would, again, cause the count to be reduced. And you're going to go into an inert growing environment, essentially. It's not, I mean, it can never be truly inert, but you know what I mean? It's like, it's basically devoid of all the things that it would find in the earth. So, in that sense, actually soaking seeds, I think, is a good idea, because with, I would, the one pushback I would say is that hydrogen peroxide is definitely not the best chemical to use, though, for various reasons, because it's quite harmful in many respects for other reasons. Um, there is other things out there which maybe we'll go into separately, but finding the right thing, I think you'll get away with just water soaking in that situation. It's fine, especially if the water doesn't contain too much minerals or salt from, you know, tap water is not a good idea, for example. I always try and use rainwater, reverse osmosis, or filtered. You know, that's basically my preference order. Filtered actually works quite well if you let it, you know, dechlorinate naturally for 24 hours or something like that. But that's my commercial answer. You want me to go into a facility and pop 1,200 seeds to, you know, put into production immediately because they're grown from seed, because that's the way the license is set up. Then sure, yeah, I'll do a 16-hour hydrogen peroxide soak at 0.5%, because it's probably in their SOPs, right? So you see what I mean? I didn't write that SOP though, because again, I would use a more natural more natural substance like chloris.
What would you use?
I'd use chloris. Chloris is a substance that your body makes to fight infectious agents. When your macrophages, latch onto a microbe, they inject it with chloris, and chloris is from the chloride family, but chloride's like very, very basic in pH, so it's got high pH. Chloris is pH neutral, and you get commercially available chloris, but it's chloride that's been pH down, and obviously there's a bunch of acid in there, right? But if you can find someone who naturally synthesizes chloris, you can spray it on your face and your eyeball and your mouth, on your clothes, does nothing, but it keeps all the bad pathogens at bay. So that's what I would use, because it also it doesn't. I don't believe adding anything enhances germination, but what that can do is stop any potential infectious agent from making the seed abort, which is something that's obviously not talked about much in cannabis as well. What they call in the other crop industry, they call them abnormals. So seeds that partly develop and then they just do something weird and often don't go beyond that point. Usually in a seed lot outside cannabis, you get anything between 5 and to 0, but usually around about 5% abnormals in the seed batch. So, but that's often because of some kind of residual microbe that makes the plant basically just go into shock really early and stop and abort development. So yeah, I would use chloris, but that's my commercial answer. If you're asking me how I would grow seeds on a balcony in Spain, for example, then those seeds would go into compost soil, and they would go on the window ledge with some damp newspaper on top of them, and they would be left there until they germinated. Some of them will come out in 24 hours. Some of them might take 10 days, but that I've had consistently 98% germination success doing it that way. Whereas in the commercial level, I don't get anywhere near that rate. Often I'll get in the 90s, but it's I do lose. And then again, if the seed's older, then pre-soaking it actually does maybe help, coax a couple of them out of their dormancy a little bit. But general, my approach would be use a good soil, and I don't know, we don't have the same brands, okay, so it's hard for me to say what you'd use, but I like John Innes #2 potting soil, very good soil for starting any plants off, and I just use that same technique that I would use in pretty much any other seed that I grow, which ranges massively, but that approach for me is just the best, and again, if you keep my idea is that I want that seed to be interacting with a natural biome as soon as possible. That's my goal there, and you let that little synergetic exchange work itself out, and then everything will be okay. It's kind of like I don't know, it's a throw the grenade in there, just hope for the best, the kind of thing. But it's consistently the best way that I've found to get high levels of germination from a seed lot. But I would honestly say there's no special trick to any of this stuff. There's no magic way that we've figured out that's scientifically better in every other way. Just take the seed, stick it in some dirt, and look after it for a bit. Keep the environment as good as possible. Again, some of the polyhybrids that we grow today, right? Some of those varieties that have been really crossed without being stabilized. Some of them have lost a lot of their hardiness. So all these polyhybrids that have been crossed and not stabilized at each cross, right? Like the Skittles line's a really good example of that. Some breeders now, I know the Peyote Skittles from Seedsman, for example, is much more stable, but the original Skittles cut, I don't know if you know about this, but people talk about how crap it is as a plant, in terms of it's really fickle, it dies really easily, it doesn't like too much this and too much that, and I think that's a consequence of constant indoor breeding and refinement through artificial environment, and somehow they lose their ability to be hardy. Whereas I've got a friend who germinates hemp seeds on his window ledge in Scotland, and it's dropping to 4 degrees Celsius and stuff like that, and the seeds still germinate. And that's something, again, one of the projects that I'm working on, which we'll maybe get to in a minute or so, one of the projects I'm working on is to try and help bring a bit more genetic diversity into the masses, and really start playing with some of these outlying genetics to really help reblend the gene pool that we're dealing with, because, like I say, there's so much incestual breeding going on in the cannabis space that we're losing a lot of the essence of what makes the plant great, which is its adaptability. And that's what I'm hoping that some of the interesting stuff will come out of your citizen project for this very reason, because I think we'll find the ranges that we thought they were growing in, and sometimes, I think if the seeds are coming from like Humboldt or something like that, the range will be much wider than they probably expect, because the way that they breed, it's outdoor and it is, cold at night times late in the season when the seeds are setting. It's cold in the early season when they're setting them off. The same. You're in Oregon, right?
Washington.
Washington, yeah, yeah. Well, so yeah, you've got like a similar, probably a bit more extreme version of that, and maybe a slightly shorter season, right? But I think the way that those guys breed and develop seeds in Northern California lends itself to naturally encourage that adaptability to
Hardiness.
the environment.
Yeah.
Sorry? Yeah, exactly.
I think it's important for people who are listening. They might not really understand, and this is something I talk about a lot, and you obviously know. But a lot of breeders today, these polyhybrids where you're talking about, are unstable genetics from inbreeding, crossing, hype strain over hype strain over hype strain that aren't showing. By stability, we mean the same characteristics, similar characteristics from like over 80% of the seeds. We see a huge range of phenotype variation. Whereas like the Humboldt Seed Company is breeding, I think, to at least F8 before they release or something.
Right.
So it's bred for stability. That means they're crossing it and making sure that it's showing the same characteristics. But they do something a lot different because they're breeding outdoors, which most breeders I know are breeding inside, and where you have this perfect little environment of, oh, I don't let my temperature swing more than 20 degrees, and I don't do this, and there's perfect this and perfect that, and my VPD is matching a chart. And then you expect these plants to, to have the hardiness or the range, but we're losing the diversity because we keep inbreeding. I mean, Skittles is in like so many things too. Like people are just crossing Skittles, Runts, cookies. It's like,
and they're all
we
in the
want to
same
see
lineage.
yeah. So you are working with Landrace Warden, correct?
I help the Landrace Warden,
Okay.
so it's more a casual relationship. But that is part of, again, a project that I'm developing. Actually, not allowed to say too much about who's
Okay.
involved in it, but what we're basically doing is we're trying to find. So the Landrace Warden is a seed brand. I'm not going to pitch them, but it's quite interesting. They're taking basically seeds from source in Afghanistan, Pakistan, places like that, and they're making them available like through Seedsman, for example. But what they've also got there is actually collections of seeds which are a little bit more rare, and they're not really suitable for putting in packs and selling because there's maybe only 100 of them, and once they're gone, right? There's a lots of generational farms in some of these little remote places in that part of the world, and they've got heirloom varieties that they use for their occupation, I guess, for their for making a living. And because of things like the 2 year in a row floods in Pakistan, some of those things got lost because they planted their backup seeds the second season and then it got flooded again. So we've ended up with little collections here and there of different rare trinkets, and we've got stuff for that part of the world, we've got stuff from Asia, we've got some stuff from Africa, and I've got an idea of making this available basically. And to be honest with you, it's very much about remunerating the people who made those seeds, and it's a lot about actually a very niche target of the audience where we're targeting the breeders. Come and buy these seeds, start with a bit of, outside genetics, and really starting to hone something different instead of always chasing almost like the same, thing. Oh, that's cakey, or that's fruity, or that, like let's widen it because it's there. There's also historic accounts will tell you there's lots of THCV dominant plants, which are now somehow not in. It's a rare thing. You get the odd little strain here and there, variety that's got THCV dominance, but actually that was much more prevalent maybe pre-90s, like when more natural genetics, or sorry, I should say landrace genetics were in circulation. So that's going to be a fun project. You'll be well informed of that as it unfolds, but we're trying to, basically create a bank of really high end Landrace genetics, and the selling point is as well, like we know that these will grow wherever you put them. They've grown them, like, really high. Yeah, they grow anywhere because the environment in which they develop is like extremely cold at night and extremely hot during the day. A lot of places. And it gives them that hardiness, that range. You get beautiful coloration in some of the ones that come out of the ones that, for example, the Landrace Warden sells just now. That's because of the environment they're growing in. It's not sometimes it's genetically triggered and the genes have to be there, right? But actually, it's a natural autumnal occurrence is coloration in the fall.
Let me kind of wrap my head around this. So you're partnering, like trying to partner or work with people who are saving these genetics from all over the world. So the Landrace is strains that have grown naturally and adapted to that environment. And so with current genetics, we have kind of a bottleneck of the same gene pool. We're starting to get a lot of the same flavors because breeders are just inbreeding with the same few strains. And so by having this wide variety of what naturally has been happening all over the world and people have been cultivating these local strains, we're going to be able to rejuvenate the genetics and kind of pull some of these more rare cannabinoids, like you said, THCV, and get some different flavor profiles, which also means different medicinal profiles. Entourage effect's going to be different, and they're going to be a lot more hardy for growing outdoors, which is amazing for our project because we want more people to grow outside.
Yeah.
So
But
that's
yeah,
really freaking exciting.
yeah. Again, it's not always going to be different cannabinoids, but it's going to certainly be, sorry, not always going to be a different dominant cannabinoid, but you're going to have different ratios of those lesser known cannabinoids and a higher percentage, within some of those plants. And one of the other interesting things is, along with these varieties, we can genetically prove that they're distinct, and we can also then have like from source descriptions. We've had some crazy write-ups on some of the effects of some of these, everything from things that smell on the plant like rotten meat, all the way through to like super unusual psychoactive effects. Like what that means, I've got no idea, but I'm excited to learn. So again, some of the compounds involved, and the science has finally caught up with something I kind of believed from the start, was it can't possibly all be about terpenes and cannabinoids, right? Because if it was that simple, then it would be easier to replicate in an artificial way. So the science has caught up with that a little bit, I think, and actually now proven that, yeah, we have all these other sulfur-containing compounds and esters and all these other things that actually contribute to flavour and taste. So what I'm really excited about is diversifying the gene pool, and then also crossing these with what is in current circulation should give a nice blend of different things. And then for your project, like we've got ones there that are specially adapted for water use efficiency, so they can survive and flourish on much less water. Simple as that. Not fancy in any way, but a super important trait if you're in a kind of drought-driven climate. There's other ones with, actually that's the same plant. It's got this like just an incredibly dense root network and an extremely long taproot, which I don't know how far it goes down in nature, but it goes far, as far as you can't pull it back up, it just keeps snapping, like one of those kind of things. But loads of things like that. We've also seen a thing like a phenomena that you would actually dismiss if you were a home grower, right? Which is after the plant starts to flower. So you just say you're doing indoor artificial lights and you've grown 18-6 for 4 weeks and now you flip it to 12-12, right? Now, what you expect to see during that, pre-flower, early flowering stage is a bit of a stretch in the plant, right? Now, what if I said that stretch doesn't stop? But the plant also bulks as it continues to stretch. So you end up with these much more traditional looking long sativa flowers kind of thing, but they're not sporadically, they're long and thin, but they've actually developed, the florets within the buds are actually developed, and it takes 14 weeks to finish that. and then that doesn't smoke or taste or smell like anything else you've ever tried before, because again, part of the big crossing is always driving the time down it takes to finish, like it's some kind of, oh, you can just preserve this and do it in 10 weeks but it normally takes 15. That doesn't actually compute very well. The science doesn't back that up very well at all. The genes that are responsible for shortening the flower time have got absolutely nothing to do with the complexity of the biosynthesis of these compounds later in the plant's life, which might actually be triggered by the death of as the fan leaves start to die off and you've got nothing but just bud and sugary leaves. You're having a massive impact on the plant's ability to take up light energy. So now you're metabolizing all the backlog of all the inputs, and how, because it goes in as light and it comes out as like bud. Like, there's a lot of intermediates in between that, and that backlog has to be cleared at some point, and that changes the chemistry in the end. That's why the drying and curing's got a bit of that going on as well, I believe. But in a 14-week flowering plant, you're going to find things like much higher accumulation of sesquiterpenes, as opposed to monoterpenes, because monoterpenes come together and make sesquiterpenes. So if you put them all in the same place over time, they build up. It's also true, but if you cut the chemistry off, that those sesquiterpenes will break back down into their monoterpenes, right? So all of this kind of intricacies, and that's just one of 240 known terpenes, and we're now saying that, well, there's at least 8 other classes of compounds involved in that kind of smell and aroma and all that stuff, and that's not even mentioning what's going on at the cannabinoid level. So it's quite a lot of things to be looking at and trying to find new things in, but the ratios and variations that already exist should tell us that what we have in front of us when we go to a dispensary is not really, it's probably only a part of the spectrum. You see what I'm saying? Like, there's a whole other realm, I think, that we've just not really unlocked yet, because it's not been intensively bred the way that these. I mean, I love some of the modern strains, like, that you can't say that the latest stuff coming out of Humboldt's not good. And another seed company I'd mention out that way would be Atlas, who do the breeding outdoor as well. They're really good. So yeah, that's that to me is I think I spoke to you about it the first time we spoke as well, about the need to increase genetic diversity. So I see this opportunity to do that, and I'm going to try my hardest, even if my part in it is just pulling everyone together and facilitating it. That for me is worthwhile, because we will end up with new things to explore, and that's exciting. And especially because I'm not like an expert breeder like Ben or Nat, right? So when they get these genetics, they'll go in all sorts of directions that I would not go in, because I'm probably following a genetic map or something silly like that. So these guys, but in saying that, they are using a lot of science in their approach now as well. I think they're doing a lot of sequencing and different things. So it's super exciting, and I think just giving these guys an option to get some of these things that are definitely outside the normal gene pool will really help diversify the full range of catalogue that each one of them holds. And if I can play a part in that, then I'll be super happy about it. But we should also think about how the Citizen Science Project could fit into that, because we could keep some of those seeds for special projects and maybe do a couple of crosses just with each other to multiply out the seeds, because although they come from source, we don't want to replicate them. We want to keep them as from source. Like that's part of the USP of those kind of land races. So if I just replicate them in Spain, like, I think it takes you a step away from that natural tendency, because those seeds are then grown in Spain, right? They've not had the same exposure to the extreme climate as they did in halfway up the Hindu Valley, for example. So that's super important, I think. And then, but the idea is you could take some of those and cross them, and then you're saying, oh, this is more grower friendly and less niche for breeders. That's the idea. Again, I don't know if it will pan out, but that's the idea of something that I'm quite passionate about now. Yeah.
So let's talk about that for a second for listeners. Like, a landrace strain is something that is stabilized in a specific environment by naturally breeding within its population in that climate. And so we've got kind of distinct what we call like the sativa and the indica, which is really more of a physiological differences in the plant rather than a taste or an effect. even though that's kind of like what's being marketed at dispensaries to consumers, unfortunately.
Yeah.
And
Yeah.
really, I think that's because they're associating just like the secondary metabolites, the esters, the terpenes that are associated maybe with different regions, which is a result of the environment, because terpenes are communication of the plant with, you know, its environment, basically, like the insects and the pressures that it encounters. So you can kind of see like replications of like flavor profiles, and that might be why we kind of got that misconception. Oh, sativa's you know, fast pacey, uplifting. Indica's like couch lock, into couch, you know.
Yeah.
So let's hear your description as a geneticist, as a man of science, of the differences between an indica, sativa, and ruderalis.
Yeah, so I actually agree largely what you said. I think the ideas have been simplified and condensed greatly. I think the range that you would find within any one of those subspecies is much greater than you would expect. I think you'll find long, thin, stretchy plants that follow the sativa body plan. I think you'll find some of them will send you straight into that couch lock, and I think you'll find some of those indicas will be very uplifting. So I agree with you. I don't think the effects necessarily are defined by the genetic heritage. And as you say as well, the environment itself plays a huge role in deciding what final compounds come out of the plant. So I've felt like I've been fighting a losing battle since I joined the industry with this, and I just says, listen, these terms are no longer relevant. I go on seed banks and they say this one's a 63.5% sativa, and you're just like, what? How did you come to that number? Like, show me the cross that led to that. And anyway, let's talk about genetic inheritance for an example, right? So when you okay, in fact, there's two examples you just mentioned. So you've got a pool of genetics which naturally are adapted to this specific area, right? And they're landrace genetics. There's lots of variation within them, but they're all specially adapted to suit this special niche, right? And they have a bit of variation which allows. That's what natural genetics do. They have variation within them, so if something happens, one of them will survive and then that will become the dominant species. It's called genetic drift. That's, it goes from this genotype to that genotype, but that genotype remains in a lower frequency, right? So you've got all of that going on. Now, if I have something that I'm calling a sativa, that's because it actually comes from a certain place, right? And that's what the original definitions were. In other species, you get japonica as well, which are things originated in Japan. So indica is from indica, and so you have all these definitions that were based on where those seeds started out. But actually, when you do the genetic analysis, you find that prior to any known trade lines, sometimes that these species had clearly been shared between those more remote areas. Okay, so and all the different people, all the different efforts to categorize sativa and indicas has largely been based on the history. Okay, right. So all of those different areas had a specific gene pool, but we've shown that actually there was a lot more trade going on there than what we thought. Now, if you look at the genomic classification of any one of these, it's subspecies, right? Which means it's basically no anywhere near speciation. You can cross them between, they're still the same species, but they've been adapted to specific niches. That's where it ends, right? So for me, when I first learned about it and I looked at it from a genetic standpoint, the descriptions are actually only, as you say, describing a physiology. For me, between indica and sativa anyway, it doesn't even go anywhere beyond that for me. Because, and then when you cross a sativa and an indica, sometimes you get a blending of some of the traits and sometimes you get a dominance from one side or the other. Now, again, we don't see much written about that in terms of this is you see a sativa leaning or sativa dominant, you get those descriptions, but there's like 25,000 genes, something like that, 22,000 genes in cannabis, right? So it's not completely dominant when you do a cross. It can never be completely dominant, one, because that's just that you need to do a self to get all the same genes to come out like that, right? So when you cross, you've always got degrees, and you've always got, well, in this case that gene's dominant from that parent, and in this case that gene's dominant from that parent, and in this case the genes are blended. So the leaf shape morphology, right, the architecture of the plant these actually, using the Indica and sativa labels for me, best describes that. And, maybe to a certain degree, bud structure and node spacing, right? A little bit, kind of secondary characteristics maybe. But that's where, for me, that's where the usefulness of those descriptions pretty much end. And I think the science largely backs that up as well, that it's not really a classification that like it's so absolute like we use it as. And then ruderalis is so funny because everybody talks about ruderalis like it's one plant. Like there's just one lonely ruderalis single plant somewhere in Russia, and everybody's taking a cutting of that, and that's how we got autos. But as a subspecies, there's numerous, varieties within that, and they all do different things as well. And it's a gene pool that's not been really explored at all. Who's not to say that some of them won't actually produce even more interesting chemicals than the plants that we've come to revere, So again, we've not played with these gene pools enough to have actually a real grasp of what's going on. I know a company that spent somewhere in excess of £20 million on genetic research, and their scope of view is very narrow, because they didn't go and look at 17 different ruderalis varieties and 17 different, ones for that. I'm just going to say now this region and that region because it doesn't matter if they're indica or sativa. The point is you're trying to get as far out from this main focus that we have on all these polyhybrids. You're trying to get further away from that because you get a much more transparent genetic backbone to frame everything. When you do this, your scope goes deep and you can tell a lot about these small things, this small group. But if you go outside that, you can't really learn much. You see what I mean? Because it's so incestual, and that's for me probably, yeah, that's my take on all of that stuff. I think it's at one point it had a really good and useful meaning, and I think now it's largely redundant. Yeah, unfortunately.
So when home growers are deciding what seeds they should choose from, and as you said, breeders put like, 63% sativa or whatever, and you're like, okay, yeah, exactly, how do they come up with that number? What do you think is important, or how would you choose genetics for your local environment? What would be your process if you're growing outdoors?
Yeah, so that's an interesting one. I think like the luxury of being in the industry is we have lots of contacts and we can speak to lots of people, right? So that's always my first thing, is like steal the wisdom from other people, because that's, it's a lifetime of experience on top of my own. So, that's invaluable. But for people who don't have that, I think it's really just about, understanding your own environment first, having a good grasp of not only the averages, but the extremes and the maxes and mins of all the different conditions, because you can't control it and manipulate it in the same degree as you can in an indoor grow. So once you know your parameters, so if you're hot and dry, for example, you have a different set of, different set of traits you're looking for than if you're the kind of dull and wet, as we are in, say, for example, in Scotland and Britain largely. So I'm looking for hardiness probably no matter where I'm going, right? So how robust is the genetic? What is the parentage of the genetics, and does that tell me anything? Because again, if you take something like, something that's quite closely related to something that's from a Landrace or something like that, then you know that it's not too removed from that kind of hardy environment to a certain degree. So, but I'm looking for, in a wet environment, I'm looking for an open plant structure, to reduce the chances of mold and help airflow. I'm looking for things like, again, in that environment, how bushy is the plant going to be? I don't want to grow a wee stocky indica plant shape in a cold, wet environment. Do you know what I mean? Because that's just asking for trouble. But at the same time, if I was somewhere really hot, then that's what I would be looking for, because actually the plant is designed to keep itself cool in the inside. So yeah, it just depends. But for the UK, that it's something I learned when I was in California, and they say if you don't like the weather in San Francisco, just wait 5 minutes or something like that, and we're very much like that in the UK as well. My friend up in Scotland's done a bunch of got a bunch of farmers to do cooperative hemp growing, and we've used a bunch of genetics there, which have been quite telling in terms of how adaptable the plant is. Um, but yeah, it's a hard one because I'm so used to, okay, this is the parameters, go and do it. But if it's personal preference, yeah, I'm probably looking for something that flowering would trigger quite early. Like flowering, well, although people believe 12-12 is the thing, there's actually a full range in there. I've seen plants flowering under even like 14 and a half, 15 hours. They'll go into full flower. And again, that can change the kind of, if you keep it at that, it'll change the structure of the bud, I think, in the end, if you're doing it indoor. But I want something that's going to trigger early and then finish quite fast, again, just to beat the extremes of the wet season that comes in here, I guess anywhere between mid-September onwards, you could just get torrential rain for 2 weeks. So that would be the things that I would be looking for. Also, something that wasn't so particularly dependent on direct light. So like some plants need like 12 hours of direct light during their veggie period, and some of them, but some plants will actually 5 and 6 hours of direct light, and the flowering time is actually enough as long as everything else is pretty good. So and again, just as I'm saying this, a variety comes to mind, which is green poison S1 from I think Sweet Seeds, which is a plant that would flower in 14 and a half hours light and finishes in 6 weeks. So perfect for this environment,
Wow. Yeah.
yeah. So those are the things. But again, I feel as I'm saying that it's very specialized knowledge. Like it's quite. I'm feeling that's actually not quite an. That's quite not a good answer for beginners. People are just trying to figure out what they're doing, unfortunately. But it is a
Well,
hard one.
I think that you have good points, though, that the listeners can pull from. So, understand your environment, your extremes, look at the lineage of your strain that you're considering. So, not only take into consideration the flowering time, which is listed by all breeders, but then go in. I like to use seedfinder.eu because they have like 33,000 strains, like the family tree lineage. So, you can see where they're pulling from, Landrace, and then kind of associate that with, okay, so where's this landrace from and what kind of environment is it and what kind of structure, like you talked about. That's why it's important to know that the sativa leaning or indica leaning, if it's going to have those characteristics, what are those leaning characteristics going to be and how's that's going to work in your personal environment. So I think your answer was great. It's just
Phew.
you have to do a lot of research right now.
Yeah, because there's so much choice, which is good in one respect, but sometimes you're dumbfounded by the choice. Like again, go back to your OG Kush. If you go into a seed supermarket and you type OG Kush, you'll probably have like 12 varieties. 12 breeders presenting an OG Kush. So that's something that's quite tricky to navigate as well, because not everything's fair and transparent in this world, and certainly not in the seed world where there's a lot of shady dealings going on. And again, I was lucky enough to work with a company that was trying their best to avoid all of that and trying to actually push in the opposite direction. And when we went, and that's why they work with like Atlas and Humboldt Seed Company and people like that who are also of that kind of ilk. And there's a whole bunch of players out there that will just do mass scale white labeling and just call it the seed packet by whatever's the latest trend. So that's something that people have to navigate as well, is they don't go to some, if somebody approaches you on social media and says, oh, you want to buy some seeds, mate, they take it with a pinch of salt. And if they're claiming to be a big seed bank, they're probably not. Go to the website, do your due diligence, find the good suppliers, or go to the breeder direct. That's even better, in my opinion, because then sometimes you will actually get access to advice as well through that, and nobody knows those plants better than the breeders, right? So you can, if you can ask them the questions of, I'm living here, what should I grow? They probably know off the top of their head what to go. But otherwise, it's 33,000 strains. Wow, that's actually, I didn't realize it was quite as many as that. But that's almost more strains than there are genes. So how does that figure, right? Yeah. But anyway, but yeah, that's an interesting one. Um, The
Yeah.
other way to do it again is probably is through asexual propagation. If you know someone who's already done that work, and then get a clone from them to grow your plants. I guess it depends if you're doing natural outdoor growing. That's a bit more tricky, but that would be another way to secure a genetic that has been proven in that environment. You've got, I think, a
Definitely.
lot more of that stuff going on over in the States now. Is clones kind of readily available as seeds or?
In California, I always had clones. I could call up five people and
Oh,
get list of 200 to 500 different, like, oh, what do you want? What do you got? I got
right,
like every flavor. In Washington, no, it's very difficult. It's actually still illegal. It's a felony to home grow in Washington state. So they don't have genetic nurseries available. Like California, you can just go buy clones from the dispensaries.
right.
Um, but it's crazy that the state of Washington is making money off of selling cannabis, but it's still a felony and like a $10,000 fine to grow at home unless you have a medical license. So, very hypocritical, Washington. I hope you're listening,
Yeah.
legislators, and your constituents aren't happy. Let's circle back then. We were talking a little bit before we started recording. What's the vibe like where you're at in the UK? Can with home grow?
I mean, but actually, so without giving everyone a history lesson, there's a little bit of what they call devolution of power in the UK. So Scotland is its own country, but it's run basically from London. Over the last 10 or 20 years, we've slowly, built up our own parliament and we make some of our own decisions. So Scotland's actually making a little bit more progress and under the radar actually than the UK proper. It feels like we're still in the dark ages in the UK. There's lots of fear and stigma attached to the plant still, and even although we've now got a meek number, but 30,000 ish patients, not all of them are acting. That's like 30 active. That's 30,000 patients registered. So it's not a whole lot in a country with almost 80 million people. So in comparison, I think that the German market is got like a whole percentage of, it's like a 1% or something like that, which is huge. We're way below that. So we've got stifling all the way through the industry in that respect. But in Scotland, we're trying to push for non-prosecution of home grow for medical users, right? Which is a huge step forward because right now, you're not allowed to cultivate at all. You need a special license from the same people who do like firearm licenses. It's called the Home Office. It's where all the kind of drug stuff it goes through as well. And they argue between themselves, the government departments, who should have right over this. For me, as a plant, it should be under the agricultural department, and that's where it should be left. It should not be part of the drug policy because it's not really a drug because it's being psychoactive doesn't make it a pharmaceutical. And that's basically what the word we use for drug is. So that it's a bit of confusing. I think confusion in the language, which is deliberately, being pushed on a plant, which has no place being called a drug, really, because it's a wellness medicine. Do you know what I mean? Like it's quite different, and it's not a single active ingredient that's causing all the benefits. We know that now for sure, even although it's still largely hypothesized that the entourage effect is true. We kind of see in the results through the trials. That's one thing that I do have privy to in the UK is a lot of the medicine that's been given out and it's been isolate has been less effective for certain things than say full spectrum. So that's interesting. So what we're pushing for in Scotland is to have non-prosecution for homegrown, which would be a huge thing because the way the police structure works there, we have something called like I think a Lord Advocate, and he basically decides what laws are going to be really enforced and which ones we're just going to leave as kind of like passive. And as long as nobody's doing anything too crazy, like growing 100 plants and clearly profiteering from it, then they're not going to get punished. So that's as close as we've got. But the UK on a whole, the Westminster Parliament in London and stuff, they're still very much terrified of cannabis and its effects, and they're still trying to keep it locked up and kept under lock and key. The funny thing is, all the fear mongering that's gone on in the press has been around like for decades. Like skunk super skunk, 14% THC causing psychosis, and now all the medical weed is all like 25%. Do you know what I mean? Like, so there's this whole big, if you lay the argument out, it's absolute nonsense, and it's there for everyone to see, but yet we can't seem to progress. Alex, it doesn't seem to be, there's so much infighting and silly nonsense, people just too focused on profiteering from it, that they won't allow things like homegrown because they have already invested and raised money for this vertical that grows its own and sells its own, and and it's just all just corporate bullshit
Bullshit.
on a large scale, Yeah.
Exactly. Okay,
Yeah.
so last question. I don't want to keep you too long. Thank you so much for spending so much time with me.
Not
Um,
at all, not at all.
you mentioned the entourage effect and how we are, you know, this is widely accepted in the cannabis community that different secondary metabolites have different effects on us and different brain chemistries. So different people are going to experience different types of highs, and it's going to affect them and help them in different ways. So you mentioned some unique cannabinoids like THCV, what, and also just experiencing different isolates. What is your favorite secondary metabolite that's produced by the cannabis plant then?
Hmm. So I've not actually. One of the things when I visited Ben at Humboldt Seed Company, he said that he had basically all the terpenes, for example, all isolated, and you could go and sample them. And they do the scratch and sniff seed packets, which I think is absolute genius. And that's one of the ways that they're able to capture those. But I've never actually been able to strip back and try all of those things independently. So I'm still somewhat confused in my own mind about certain things I like over what's actually causing that. But as far as the full range concerned, like, I've found CBG a really interesting cannabinoid because it doesn't have a psychoactive effect, but it does create some kind of wave through your body, right? I don't know if you've experienced CBG. Have you tried the CBG flower?
I have, well, not CBG flower, but I take mushroom gummies that have CBG in them.
Right, right. So having tried CBG flower, I was amazed by the effect, and had a calming nature without the high. It was so weird. My body couldn't. It's like, what? Like, I should also feel like this, but no. I just felt relaxed. It felt like it lowered my anxiety. I didn't feel so. What's the word I'm looking for? So I didn't feel like things were so needed immediately addressed. You know. But again, I wasn't high. I wasn't like under a hit. It was like it was very weird because I had all the calming effect without any of the psychoactive effect.
How can you, since CBG is the mother of all cannabinoids, right? It's like every other
CBGA.
cannabinoid. Yeah.
Yeah.
So how do you get a flower that's high CBG? Just cutting it early, or
Oh no, no.
what's?
So that slight misconception is CBGA is the precursor to
Yep.
CBG, THC, and CBD. So actually, if you don't have an active THA synthase gene or a CBDA synthase gene, then all of that CBGA goes straight to CBG. So you
Okay.
do get CBG dominant plants. CBG is the one that's like, yeah. So I'm sure it's just one. I'm sure it's just one catalytic step after CBGA, which in the other cases it's two or three for THC and CBD. So you have potentially 2 points of mutation there, two genes to target to knock that pathway out. Do you see what I mean? So it's actually it's a bigger breeding target than you would think, because actually
Okay.
knocking CBG out is much easier, much harder, because it's one gene that's responsible for that conversion. So yeah, it's again, it's a, if you look at the wider gene pool, you'll find more CBG dominant plants there naturally occurring because those mutations happen spontaneously and they shut down the pathways that make THC and CBD. So you're left with only CBG essentially. But you also get ones where you've got partial activation of the synthases, which means that you get different ratios, and different through different individuals. Something that from all accounts, it's hard to stabilize in cannabis genetics is consistent ratios. Like even a CBD to a THC plant, 1-to-1 within a seed pack, you tend to get 2-to-1 to 4-to-1 variation. Really, really hard to stabilize, as you rightly point out, because the environment has such an effect on the plant's secondary metabolites. But yeah, CBG is the one that I think would be interesting for people who want to try cannabis without getting high, which, again, maybe more medical users and stuff. And I don't know what CBD is doing in the US, but here it's doing weird things where it was falling under novel foods. So the Food Standards Agency had a big role in it, and they've said that toxicity levels is and they've dropped the allowance right down to 10 milligrams or something like that, where it was before it was 70. And it was and even that was for me 50% low where it should below where it should have been. Based on, as far as I can tell, based on a singular rodent study where they looked at liver damage in rats who were given the equivalent to 70 milligrams per kilogram of body weight, and again, having worked in various research centres, I know that doesn't always translate to higher being studies. It doesn't. Those rats have got very different metabolites from us. There's lots of similarities, but they can withstand much more extreme exposure to waterborne diseases, for example. They live in the sewers, where if we went down there we would get sick as hell. You know what I mean? They don't. So there's a completely different biome, it's a completely different organism, and sometimes it translates well. If you're looking at the individual cells and how things work, how things get through into the through the blood-brain barrier or something like that, it translates really well. But things like your metabolism and how you process metabolites, that varies massively because your diet can influence that. And and that's what I was going to say is, when you're talking about all this variation, one of the things that I notice you can control the accelerator a little bit is whether you fast or not. If you consume cannabis after 16-hour fast, it's going to feel much more impactful, than if you've just been eating all day and then you have it on top of that, and because it's slightly different things. But anyway, so I think CBG is interesting for that reason, and for at least in this conversation, I'm going to say that's my favorite secondary metabolite.
All right. So last question. Do you got the details? How do I get THCA dominant flower? Because there's a loophole going on right now in the US where you can sell THCA and ship it across state lines. You just can't do higher than 0.3% THC. And I don't understand how you're getting flower that has dominant THCA with less than 0.3% THC. I get it for isolate and going through hydrocarbons, but how are they selling flower?
Yeah, that's a strange one, because I've seen so many of the companies I've helped out have tracked THC levels week by week, right? So THC, actual THC delta 9, usually is never much more than a couple of percent by the end of the harvest, right? And it's all THCA because that's what the plant synthesizes, and then it naturally decarboxylates, right? But that's actually why you burn it, and it becomes, you know the story. So to get the THC levels down below 0.3 whilst maintaining high enough THC levels, I guess, yeah, cutting earlier would probably be an option. Not drying properly and certainly not curing, because all of that is, it's a function of time. It's the same as somebody once asked me, how do you do a CBN plant? And I was like, well, it's just a THC plant that you've left behind the fridge for 9 months.
Last year's depths.
Yeah, exactly. So I think it's a wee bit like that. To be honest with you, I've never seen or even thought of people doing that to get round the law. I didn't know the law was that malleable, because the way it's implied here, it's it's that compound plus any of its, like, isoforms, mimics, anything that kind of fits that rough shape, biochemistry wise. So it's much more broad, So like if that compound goes in, all its cousins go in as well, is basically forbidden. So, but I know that you guys also had the Delta 8 thing going on for a wee while and
Uh-huh.
various other things like that. So I don't really know, to be honest, but it sounds like an improbability, because unless you're controlling yeah, so maybe you're growing a plant, it's THCA dominant, as most plants are. It gets to a certain point and you change the atmosphere in a way that prevents decarboxylation. I don't know, but as I'm saying, it sounds like a pipe dream. Sounds like something, at the bottom of a mushroom
Yeah,
chip.
I don't get it. I don't
No, I don't
understand
get
how
that.
they're doing it.
Unless it's a
It's
dodgy
crazy.
COA, that could also be true, Um, because that was a
Right?
weird
Yeah.
thing about the hemp bill, about, like, just if your COA says this, so all of a sudden you could sell high THC seeds because the seed itself didn't have high THC, then it passed the COA, and it's like, what? That's not, yeah, it's either really clever or really stupid way of writing the law, and that's what it sees me always says. So yeah, that's a weird one. I need to look into that, Alex. I wasn't really aware that was
Yeah.
yeah, that
It's
was a thing.
crazy. It's
Yeah.
popping off all over in like North Carolina, South Carolina, Georgia. I've got friends who are starting little companies and able to ship from California all their isolate and flour, and it's being sold in freaking like gas stations. You can just sell it right there because it's the loophole. It's legal. It's not THC. It's THCA.
What?
I know.
That's insane. That must open up so much opportunities, but I'm not sure it's. Yeah, it just seems like more misinformation, though. Do you know what I mean? It's like kind of overlooking the actual what's actually going on, which is not ideal. Because that was the one thing I was going to ask you, is like, through all the people you speak to, and all your own experience, like, I've had to deal with my fair share of bro science, which I bought into at the time, and then later realised was, I mean, how do you go about deciphering what's real and what's not? Because just before I let you answer that, sometimes what people observe is really true. They absolutely are reporting the truth, but then they put some weird rationale on it, and it destroys what they're actually observing. So I often find that actually when there's a bullshit story, the observation actually probably holds true, but why they're saying it's happening is the thing that needs to be scrutinised. So what's your experience with all that
Well,
kind of
that's
stuff?
the key right there is the why. I am always asking why. So if there's a bro science, I'm going to go down the rabbit hole of why are they coming up with this answer, and then where does that lie, because like you said, it typically lies in some truth, and then there's a kind of divergence.
Mm.
So I think that I am very analytical and I love researching. I love reading. So I will take some information and I go to Google Scholar, and I have a great network of people to reach out to and talk to, and I explore it and I think about it and I read about it, and then I write about it, because writing really helps me get my thoughts out and really figure out what's going on, and then just keep asking why. Why would you
Mm.
do that? What does it make sense? Um, and the more data you collect, the more you can say, no, that's fucking bullshit. I almost got sold, like, some guy came on to my Patreon podcast and was like trying to sell this drops of balance, this product that was like, oh, it can do everything, it gets rid of glyphosate, it increases cannabinoids, increases yield, it kills all mold, and he was just saying like all these crazy things, and it's basically just sulfate minerals. It's just like a wide range of sulfate minerals. And so at first I was just like, well, I wanted to hear more. He didn't give me like clear science. And then I was like completely against it because I thought he was just a freaking snake oil man. But then I went and I did the research, and I started researching sulfate minerals and how they can passivate heavy metals and how they can do certain things because of the bonding that sulfate will break, and it'll maybe bond and hold on to something so the plant doesn't pull it up. And so I saw where he was getting some of these little bits of scientific truth, but then that, sales pitch and the extrapolation of saying it does all of these things, and that's where you get the bro science. It's like, okay, so maybe there is a little nugget of truth in there, and you just really have to do the research and read, which most people are lazy and don't want to read.
Yeah, absolutely. They want it all condensed and simplified down so they can get it in a sentence, which is, most things are infinitely more complex than that. And it's one of the problems I've had with writing as well. And the people say, oh, it's a bit highbrow, it's a bit over people's heads. And I was like, yeah, but that's the nature of that subject. It's complex, I can simplify it down and I can make it so a kid can understand it, but you're losing a lot of the essence of the detail, which is actually, to truly understand it, you need that detail. It's okay to say, oh, DNA is like a piece of string, but, at some point you're going to run out of things that you do with string that, what attaches to string self-replicates in a slightly different form and then, makes all these other possibilities happen from that. The template, genetics is intrinsically, complex, and all the dumbing down in the world doesn't do it any favors, actually. Make it readable so people can read it, and they might need to read it 3 times to understand it, but that's, I kind of started drawing a line under, I'm not going to go below this, this, because we're now losing the nuances of the detail and, and the pursuit of making it more digestible to people who are lazy, and can't be bothered doing the work. So yeah, 100%. That's doing the research is a big part of that. That's absolutely right.
There was a light, let it come in, choosing her sin. Every morning, choosing to be sad.